kind of interesting though to like map your cortisol onto like your So I mean, this is like one of the themes of the entire semester, right? Is trying to like correlate the subjective, our mind, which we can't. I don't have a way. I wish I did. I wish this was Star Trek or something, and I could like take your brain and like just pop it out here so we can all like see it on the screen. I can't do that, right? But what I can do is I can hook things up to your body and measure stuff, and then I can get you to observe your mind and report on it, and then correlate that with the things that we can measure. We've done that in different ways this semester. And the cortisol was one of them. We're trying to like see the correlation between this scale that you've been using all semester. What I think would be interesting would be if we had a ton of these strips and just everybody did that all semester long and see how well your cortisol matches with a 3. Is a 3 actually this value? Is a 4 actually that value? Like, can you predict your cortisol level by this scale? Is a 3 always the same, or are there things that perceptually change what you think a 3 is? So anyways, this next lab that we're going to do on the 29th with Jin Yi, has anybody seen her in the counseling services office? She's really sweet. Yeah, you met her? She's great, yeah. She leads the meditation group on Tuesdays, sometimes. That happens right after our lab. And she's going to come in and do some meditation activities with us. And hopefully they'll be interesting and useful. But what we're going to do is hook up all this gear to your brains as we meditate, and you're going to meditate and observe that experience of meditation, all right? And then afterwards, you're going to write about it and try to say, for the first 5 minutes, this happened, and then this shift happened, or, whatever. My focus was wandering, but then there was this other realm. I don't know, whatever it is. You just come up with a subjective description of your experience and try to map it onto the half hour or whatever it is sit there. And then we're going to look at the EEG that we record, and we're going to see, does your subjective experience map onto this EEG that we recorded? Can we pick anything up in the EEG? All right, so that's where we're heading next. But to do that, EEG is not trivial. I think we are going to be the first group on campus to ever do anything even marginally quantitative with EEG. They have done some EEG in the physiology labs, but just as a proof of principle, never any kind of direct quantitative. So this is going to be a new thing for us here. And I'm not even going to pretend like I've ironed all the bugs out of it. That would be taking a lot of the fun out of it for you guys. Today is an actual experience of real science. So science in the real world, the way it works is you have an idea, you have an understanding of a system, you have an idea about some implications from that background understanding, you form a hypothesis, and you want to test it. And to do that, often you have to design an experiment, and you got to get the equipment set up. You got to get the protocol in place. You got to develop. So there's one thing to come up with the abstract experiment, which I just told you is going to be, you're going to meditate with an EEG on and then write about it afterwards and see if that correlates at all with the EEG. That's fine, but there's a lot more details to that have to be worked out in any kind of real experiment. And that is where the rub is in a lot of science. It can be a major pain in the butt trying to get the equipment to work or figure out how to visualize things or how to, exactly what the right protocol to use is. It's often trial and error. So this is going to be a little tiny taste of that, not that big. You already had a little taste of it a couple weeks ago with the pranayama lab. We're going to have another taste of it this week. We're going to try to get the EEG to work and see if we can come up with some visualization, optimized visualization protocol software. There's like a ton of stuff out there. Rather than have me spend a month trying everything in my spare time, playing with my kids on a Saturday, we're all going to have fun in lab today, seeing if we can explore different ways. My mind is open. I'm not committed to anything, but I have a number of leads that I can. So before we get to that, let's talk about what EEG actually is. Does anybody want to offer a description of it before I give that to you? What are your thoughts? What do you guys think EEG is?
It's like EMG, but with the brain.
Great. Yeah, that's a pretty good description. What does that mean, though? Like, I'm not flexing my brain, am I?
No, it's measuring the like neural activity, like the nerve, like synaptic, like little like.
The firing. Yeah. Any other thoughts on it? How do we catch like a single neuron fire? Do we? No, we don't. We're catching correlated, coordinated bundles of firings. So let's see here. Oh man, that didn't come out very well. This projection. So the first EEG principle was demonstrated by this guy here, Richard Caton, in like 1870. So here, the first mention of EEG was in 1875, and yes, I did go to the literature and dig out this 100 and almost 50-year-old paper and put it here. So let's read it. I'll read it to you. After a brief resume of previous investigations, the author gave an account of his own experiments on the brains of a rabbit and a monkey. The first EEG was in a rabbit and a monkey. The following is a brief summary of the principal results. In every brain hitherto examined, the galvanometer has indicated the existence of electric currents. The electrical surface of the gray matter is usually positive in relation to the surface of a section through it. And poorly, yeah. Feeble currents of varying direction pass through the multiplier, which, when the electrodes are placed on 2 points of the external surface, or one electrode on the gray matter and one on the surface of the skull, the electric currents of the gray matter appear to have a relation to its function. When any part of the gray matter is in a state of functional activity, its electric current usually exhibits negative variation. For example, on the areas shown by blah, 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 rotation, head, and mastication, which means chewing, negative variation of the current was observed to occur when those two acts respectively were performed. Impressions through the senses were found to influence the currents of certain areas. For example, the currents of the part of the rabbit's brain, which blah, 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 had shown to be related to movements of the eyelid, were found to be markedly influenced by stimulation of the opposite retina of light. So that's what papers used to sound like and look like. That was the entire thing right there. I read you guys a whole paper. And he demonstrated a few things here. First of all, that there is some sort of electrical activity that's happening on the surface of the brain, on the gray matter in particular, that is correlated with various activities. So you move the eyelid on one side, and it corresponds to some activity on the other side of your brain. So demonstrating the principle of fiber crossing, right? So fibers, sensory fibers cross from one side to the other. That's not really relevant to what we have here. But also demonstrating that it's a good thing to not be a rabbit and have the top of your skull removed so that some guy can stick an electrode on it.
Hey.
Yeah. The first demonstration in humans was from Hans Berger who lived just a little bit later. And this is actually a replica of the very first trace. That is the first EEG that was ever taken. This is an alpha wave, a 10 hertz, an alpha wave, and on the bottom, this is a 10 hertz signal. So this is like a timer. This tells him for every beat here that's 1/10 of a second. So we'll get to that in just a moment. Like hertz, it's hurting my brain. Is that what hertz means? No. Here's what a hertz is. Does anybody not understand what a hertz is? Well, I'm going to tell you anyways. Yes, a hertz is a per second. A hertz is a beat per second. Yeah. So
is that like radio, like wavelength, or
any oscillating phenomenon, anything that's repetitive. So like there's a certain frequency that I'm hitting my fist into my palm with, and that frequency is measured in hertz. So that would be hits per second. I'm about maybe 0.7 hertz right now.
as well as like the sound traveling from the clip?
Well, that's a different thing. You know, there's like the frequency of the pitch, but that's not what I'm talking about. There's the like frequency of an activity. So for example, this dot is beating every 2 seconds. There it goes. There it goes. There it goes. And hertz is simply the time between events. So here's the peaks. This is time along here. It's that time distance, time between peaks, inverted. So it's essentially beats or peaks per second, right? So if something takes 2 seconds to go from one beat to the next, the frequency is 1/2, which is 0.5. So the frequency of this is half a hertz, 0.5 hertz. Whereas this is beating every second, 1, 2, etc. And so it's 1 beat per 1 second. Is this making sense to you?
Mm-hmm.
Yeah. Is this tedious for everybody? And then if it's every half second, so this is every half second, it's going to be 2 hertz. It's going to be 1 per every 0.5 seconds. Good? Good to go? Hertz? So as we're compacting this, the frequency is going up. The hertz are going up. All right? But the time distance between them is going down. Wavelength. Wavelength is akin to time here. All right. So we go from rabbit skulls and Hans Berger 150 years ago to, in 2007, this company NeuroSky released the first affordable, commercially available, consumer-oriented EEG, along with some game called NeuroBoy that you could control with the EEG signal off your brain. You were able to literally... There's been a host of different games like this, like some Star Wars game that makes it seem like you can make stuff levitate or whatever by controlling it with your cap that's on your brain. Whoa, dude, isn't that a cool thing? Yeah. Yeah, so for example, this is the first of those. This was the NeuroBoy game that came out over a decade ago now. And OpenBCI put their gear that we're using on the market, I don't know, like 4 years ago. So that stuff's been out there for a while. What the hell is this? What is it? All right, so the biological origin of the EEG. To really understand what it is that we're measuring, I got to talk just a little bit about the architecture of the gray matter in the brain. So all these higher order cognitive processes that I talked about a bit today in lecture, they happen in the gray matter in the cerebral cortex. And when you have, so gray matter, to remind you, is the cell bodies. White matter is their axons, the tracks. So gray matter is cell number, and white matter is connectivity, right? That's the way that I was trying to explain. And there's different, you don't need to know, I put all this on there, but you don't need to know all of the layers of the gray matter in the cerebral cortex, but there are several different functional layers. And there's these different types of cells, mostly different sizes of pyramidal cells. So when these cells fire, there's going to be an electrical potential associated with that neuron. But the EEG is in no way capable of picking that up. So these cells are far smaller than you would be able to see with the naked eye, right? And not enough of an electrical potential is going to be generated for one of these electrodes to be able to pick it up. Even if, for example, I were to volunteer, because I would never have anyone in the class do this, but have you take a chunk of my skull off and stick an electrode right on the brain, even then it would not be a strong enough signal. But we have to get through hair and skin and bone and dura and all kinds of other stuff in between. So what is actually being picked up is this summation of synchronized cells, thousands of them, likely millions of them. Millions of cells that are firing in synchrony. They're firing together. All right? And they're firing together with a similar spatial orientation. They're firing along a tract for some reason. And because of that, the electrical field that they're generating is large enough that one of these very sensitive electrodes can pick it up. So we can pick this up in the gray matter here because these pyramidal cells, the organization of the tissue layers in the gray matter in this cortical surface is highly organized. It's very orderly in terms of its layout. And so the EEG signal is fortuitously easily measured from the surface of our skull. Right? There is stuff going on deeper in the brain, but we're not picking it up, sadly. We're not going to pick it up because the EEG can only penetrate just a little bit through the skull into the surface of the cortex. All right, so that's the origin of the EEG. What is the EEG? Well, they're brain waves are what we're measuring here. Rhythmic voltage changes that are going to be a signature of this synchronized synaptic activity that's happening in the gray matter. That very superficial layer of the cortex, just the top few millimeters of the cortex is what we're picking up. It's stuff right on the surface. So turns out there are 4 types of brain waves: alpha, beta, theta, delta. We'll talk about what they mean in a little bit. And they are distinguished from one another in terms of their frequency. All right, so you can see alpha and beta have sort of higher frequency, and the theta and the delta have slightly longer frequency. But also the amplitude. So how high of a spike are we getting? A peak are we getting? People use EEG for all kinds of stuff. So it's useful for studying meditation, for example, and it's used. Anyone done a sleep study before with one of these? Has anyone ever had an EEG before? Yeah. A couple of you have. Yeah. So it can also be used for pathological problems. Various types of chronic brain disease, like epilepsy or something like that. Metabolic abnormalities can be demonstrated with it. If you have a tumor, EEG, actually, it sounds humorous, but it's great for diagnosing death. So the clinical definition of death is the absence of brain wave pattern for some period of time, right? So that is the clinical definition of brain death. Well, I mean, a cadaver, the word cadaver implies that it's already been established that thing's dead. But yeah, somebody is in the hospital and they're like comatose or something like that, and then they die. The like definition of that is not that the heart, like, because your heart can stop and you're not dead, right? Your heart can stop and you still got brain activity going. Or people drown and they still have brain function going. The definition of death is when the brain stops functioning because ain't no coming back then.
Can you have cut off their head? Their functions?
Well, those are autonomic functions, so that's not something their brain is conducting. They're like reflexes that are going to their spinal cord. It's just like if I were to bang on Molly's knee and she kicked me in my shin. That's not her brain. She's doesn't want to kick me.
What is theta?
I don't know. I wish I knew everything, but I don't. All right. Okay, so this is what you're all going to look like. No, we don't have that many, fortunately or unfortunately. But it can be used to diagnose, bad stuff that happens. So this person, whoever this is, I don't know if it was him, but this person had a seizure, and here they are. They've got each of these traces corresponds to one of these electrodes on this person's brain, and they have signifiers that get mapped onto the brain or whatever. And they're all going along, and he's thinking about, Doritos here, and, being done with school, and the rest of his brain's not doing much. And then next thing you know, somebody turned on the strobe light, and his brain blew up, and he goes into an epileptic seizure.
God, they did this on purpose?
No. This is a person who probably had a seizure disorder that was having a seizure study done on them, and they had a seizure while they were hooked up to the machine. Yeah. I'm. Please don't think that I'm making light of that seizure. It's horrible. But this is what it looks like. You start to get this wild, spiking, uncoordinated activity in the brain. All right, so let's unpack the different waves a little bit. Alpha wave. The definition of an alpha wave is let's start. Before we get to alpha, let's actually start with delta waves. Delta waves are the slowest, the lowest frequency, meaning the highest wavelength, right? These like long waves, delta waves. This happens when you are in slumber. This is the sleeping, deep sleep, the delta wave. The next one, yes?
Only in adults, though?
No, children can have delta waves too, but their sleep is a little bit different. I'm not like a neuroanatomist or sleep specialist for children, so I'm not. But next, a longer one are theta waves, and theta waves can indicate a number of different things. They can mean drowsiness. If any of you are drowsy, you may have some theta waves happening in your brain. It can also be if you are awake under some sort of emotional stress, can be indicated by a theta wave. Although it doesn't have to be stressful. So theta waves are often associated, depending on where they occur in the brain, they can be associated with daydreaming or some sort of creative process where you are just trying to de novo come up with something weird, or meditation also is associated with theta waves in a certain part of the brain. All right, next one up the chain are these alpha waves. So theta waves and alpha waves are right next to each other in terms of that. Put them in order because they're in alphabetical order here, Greek alphabet. But in terms of like the frequency, alpha and theta are right next to each other, and that's a good thing because that's where we want to be, right? This is also the meditative state. This is highly indicative of the meditative state. So the alpha wave is when you are sitting somewhere restfully zoning out. You have climbed the top of the mountain, a beautiful day of hiking. You are like just blissed out looking at the clouds, not really thinking about anything. And the alpha waves are undermined when you are focused on something like reading or typing or doing some sort of like concentrated mental task with your eyes open and you are engaging in the external world. All right. Highly indicative of meditation and hypnosis or non-arousal. And then lastly are the high-frequency beta waves. And so this is what happens when you are sitting there trying to take notes, for example, or any kind of highly engaged cognitive mental function where you are like writing, taking a test, building something, associated with extroversion, concentration, logical thinking, and active conversation. So much of your waking life is spent in beta wave. All right. But our brains need all of them. And we're going to try to identify them. So first, let's dive a little bit more into theta waves. During meditation, theta waves are the most common when you are in a deep meditative state. They're the most common in the frontal and central parts of the brain. And these parts of the brain, we didn't get to the default mode network or the active focus networks in the brain in lecture today, but we will. These parts of the brain, the frontal and central parts of the brain, are associated with control, so like monitoring aspects of other mental processes that are happening. So when you see these theta waves in those regions of the brain, it's meant to indicate deeply relaxed attention or a process whereby you're monitoring your experience. Theta waves are hard to elicit. They tend to happen much more in highly experienced meditators, which I don't know if any of us could name that. So these parts of the brain, the frontal and central parts of the brain, they are responsible for focusing the brainstem, midbrain, posterior portions of the brain, parts of the limbic system. They're responsible for tuning them in to physical relaxation during meditation. This is when you're going to see those theta waves come up in control centers of the brain. So alpha waves, as I said earlier, indicative of wakeful rest. These tend to be most abundant in the posterior portion of the brain, and particularly in meditation or simple rest. So alpha waves are more easy to elicit, and they're the ones that I'm hoping that we can find. Unfortunately, they're in the posterior part of the brain, which is a little bit harder to get a signal from. But we're going to try. All right. So your amount of alpha wave goes up when you shift from that sort of goal-oriented mindset where I'm trying to achieve something. I'm like doing a math problem. I'm writing something down. I'm thinking about what I have to do. I'm like goal-oriented. When you step out of that, that's when you begin to get the these alpha wave signatures, particularly in the posterior. So that's what we're looking for, alpha and theta waves. And it would be cool to see if they correlate with any of your actual experiences or not. Maybe you, as you sit and meditate, you're like, oh my God, I couldn't stop thinking about my dog eating leftover turkey at last Thanksgiving dinner or whatever. And you'll see that you have a bunch of beta waves or whatever. It'll be interesting any way you slice it. So here is some actual hard data that shows this. What's up? Yeah. So this is meditation. This is rest. And this is over the entire brain. Is that right? Yeah, so this is from all three regions of the brain, the frontal, the central, and the posterior portions of the brain. During meditation, we see an elevation in theta and alpha waves over beta and delta. Beta and delta is about the same occurrence, whereas we have an intensity of a little bit elevated intensity of theta and alpha waves during meditation. This maps discretely onto parts of the brain. It would be cool to see this. The alpha waves are the ones that turn up the most during meditation. They're the easiest to elicit, and in a group like this, they're going to be the ones that we're more likely to see. I don't know if anyone is able to get to the theta stage. I don't know. Maybe we will. I don't know at all. But you can see here that they're the most common in the posterior part of the brain. So, like back here, above the ear, back in this region somewhere. But also in the other parts of the brain as well. Whereas theta was elevated in the frontal and central regions of the brain. So that would be, like any of this region around here would be frontal, and then central is going to be hard to get because we're going to have hair in the way. Unless anybody wants me to shave their head a little bit, I can.
A razor?
Yeah, just start with some scissors. So I think that's it. So last, the goals for the lab. Develop a protocol for recording the EEG using the OpenBCI chip. Identify the ideal electrode placement for recording EEG in frontal, temporocentral, and posterior portions of the brain. So my thought is these BCI chips can give us up to 8 channels, right? Or is it 4? I don't know. We have to look on there. But we could get at least one in the back, maybe like part the hair and stick it in there somehow, get some in the center, and maybe 2 in the front or whatever. You guys will have to figure that out, futz with it, and see where you can get the signals. As per the Lagopoulos paper, which is this paper that I pulled all this data from, it's on the Moodle portal, so you can refer to that if you want. And then develop a protocol. Once you've gotten a recording and look, wow, we're getting some kind of coherent, I'm seeing squiggles. I'm getting brain signals, EEG. Okay, well, let's record it and visualize it in some way so we can analyze it and say, wow, here's the power spectrum of the alpha wave. Here's the power spectrum of the theta wave. Here's beta and delta, all right? So and then be able to look at that as a function of time. So we can pull out a half-hour trace or something like that. You won't do it for a half hour today. You just do it in like 5-minute intervals or whatever. But pull it out in a half-hour thing, be able to look across it, and then write in your journal about what your experience was, and then be able to look back at the trace and draw some sort of conclusions from it. Does this make sense? If anybody has like a better experimental design, like a better question to ask than whatever I'm asking here, please venture that, because this is all just like protoplasmic here. All right, enough of me talking. Are there any questions?